morning, everyone. Welcome again. Today we're going to talk about miRNA synthesis and function. So after today's class, we will be able to describe the main pathway for miRNA synthesis, also to predict the effects of knocking down miRNA synthesis enzymes, as well as design an experiment to test miRNA binding. So last week we were discussing the role of the untranslated regions in gene expression. And one of the parts that we discussed the most was the 3' untranslated region. We came up with the hypothesis and conclusions about how the 3' UTR regulates expression, how it can bind factors, how it can positively or negatively affect expression. And then we discussed a little bit of a paper that you guys had as an assignment, in which you had to do 3 different things. You had to read the abstract and introduction for this paper that speaks about amyotrophic lateral sclerosis, and in particular the FUS gene, which stands for fused in sarcoma. It's a gene that encodes for a protein that is an RNA binding protein, and that seems to be increased in a lot of cases of amyotrophic lateral sclerosis. So you have to read that, and then you have to go into figure 1, which is this one, and analyze it, and answer 3 questions that is sent as a poll. So let's see what you guys answer using either your phones or your computer. First question was, how does the 3' untranslated region regulate fuse expression? The second one was the effect of a mutation in the 3' UTR in fuse expression. And the last one is, what is the hypothesis of this paper? So, for how does the 3' UTR regulate fuse expression, we got the 3 groups of this class respond the same. That is, it represses fused mRNA and protein. So let's see how do we see that in the paper. So if we look at this figure, these are the 3 constructs that the investigators use to transfect cells. And in this case, they are removing the 3' untranslated region, which will be the white box in this in the graph. And we compare it to the construct that has the 3' UTR. So when we compare the control with the 3' UTR versus the one without the 3' UTR, we see that there's a significant increase in protein expression and mRNA expression that indicates that when the 3' UTR is there, it's actually repressing expression. So very good, everyone of you that answered that question. Good job. Let's see what happens to the second question. It talks about the mutation at the 3' UTR. It's a change in the nucleotide G by an A at position 48. And we discussed a little bit last week about how is the mutation in a region that does not encode for the protein affect the gene expression, and what happens in this paper. So here are some of the answers you guys tweeted or sent. It says alters fuse mRNA and protein. It says increases fuse mRNA and protein, affects fuse mRNA, and affects mRNA and protein, but not as much as the 3' UTR. So I see that we all agree in the fact that a single nucleotide mutation in the 3' UTR of this gene is enough to affect expression of the gene. So let's look at the graph. This is the construct that has just one nucleotide change in the 3' UTR. When we compare it to the wild type that has a complete 3' UTR, we see an increase in protein expression and increase in RNA expression. The change in expression doesn't seem to be as much as the change it is triggered by just removing the whole 3' UTR. So that was in one of the comments that I received. So very good. Let's go to the last question. What is the hypothesis of the paper? So your answers are ALS patients with the fuse mutation having pair binding of miRNA 141 and miRNA 200 resulting in increased fuse protein. I guess that was copied from the paper. The second one says the mutation G48A at the 3' UTR fuse gene can affect binding of microRNAs. And the last one says a human fuse mutation that leads to increased protein expression and was identified in ALS patients with severe outcomes, maps to the seed sequence recognized by miRNA 141, miRNA 208 in the 3' fuse. Wow, that was a long one. So I see that in common, we all identified hypotheses that had to do with microRNAs, that had to do with increased protein expression, and one of them mentioned something called the seed sequence. So all of these terms is what we're going to discuss today in this class. So I'm glad you heard about them a little bit. Has anybody ever heard about microRNAs? Okay, I see some hands. Great. Okay, let's talk about microRNAs. MicroRNAs are small RNAs. The range is between 21 and 25 nucleotides of length and are non-coding. What does this mean? Anybody? It means that they don't encode for any proteins. They're RNAs as the long mRNAs we discussed last week with 3 UTRs and 5 UTRs. So these ones are so short that they cannot encode for proteins. However, that doesn't mean they don't have any function. The function that has been associated with microRNAs the most is repression of gene expression. How did they identify this? In 1993, a group of investigators was knocking down genes in C. elegans, and when they knocked down the gene called LIN4, they realized that there was an increase in expression of a lot of genes. So when they went and mapped this gene and sequenced it, they realized that it was such a short gene that only encoded for a small RNA. So that was the first time investigators ever talked about microRNAs. And it only took 7 years to discover the second microRNA. And ever since, laboratories have studied microRNAs not only in C. elegans, but also in different species. And so far, as for now, we have more than 2,500 microRNAs identified in humans that regulate thousands of genes by identification of sequences in the 3' UTR and binding by sequence complementarity. All the microRNAs that are identified so far in all the species are in this website that's called the miRBase, and one can find there the sequence, can find their papers associated and whether these microRNAs have been found in conditions, gene expression, or diseases. So, how these microRNAs are synthesized in the cell? As I mentioned, they're encoded by genes. So there's a polymerase in the nucleus that will transcribe this DNA into precursors of microRNA. The first step is a long mRNA that has several secondary structures that look like hairpins. These are called the primary or pre-microRNAs. These are further processed by a complex of several enzymes, of which the most important is called DROSHA. DROSHA cleaves these primary microRNAs into precursor microRNAs or pre-microRNAs that are recognized by an enzyme called exporting 5 and translocated into the cytoplasm, where they are cleaved by another RNase called dicer. And dicer, the cleavage by dicer results in these double-strand small RNAs called mature microRNAs. These have two strands. One is the leading strand and the other one is the accompanying strand. And the leading strand will bind to the RISC complex. The RISC complex stands for RNA-inducing silencing complex. And the major component of this RISC complex is a protein called argonaute. And when argonaute and other proteins bind to the microRNA, they recognize the 3' untranslated region of the gene and repress translation or degrade mRNA. We're going to discuss a little bit more. So I want to now do an activity with you. We're going to go over all the steps we just learned. And what I have is the strip sequence that has all the different components of the pathway and the enzymes. And what I want you guys to do is to put them in order, and then we're going to discuss them all together afterwards. So, okay. Okay, let's see how do we do. Let's put together the microRNA biosynthesis pathway. So, what's the first component of it? Sam?
microRNA encoding genes?
Okay, great. We have the microRNA encoding genes in the DNA. What's the enzyme that transcribes them? Susan?
The RNA polymerase II.
Good, good. And what do they get transcribed into?
Pre-microRNAs?
Pre-microRNAs, very good. What's next?
Drosha.
Drosha. Drosha cleaves them into?
Pre-PRE microRNAs?
Very good. And what happens to this?
The exportin 5.
Okay, translocates them into the cytoplasm, and then
Pre-microRNAs.
they get
And then the
processed
enzyme
by
dicer.
dicer. Great. And then?
You have the mature microRNA.
Very good. And these now we're going to target the genes. So what
Argonaut.
needs to happen?
Argonaut.
Argonaut needs to bind to them, forming the RISC complex. Very good. Very, very good. Good, guys. All right. So, now that we know the pathway, we're going to go into a second activity in which we're going to think about what happens if any of these enzymes is absent from the cell. So, we're going to work in groups, and we're going to predict the effect of knocking down of dicer, drosha, or argonaut that we just learned what they do. And what happens if we knock down any of these in the levels of all these different things? So one would be the pre-microRNA levels, the other one would be the pre-microRNA levels, the mature microRNA levels, and what will happen ultimately with the genes that these microRNAs target. So you're going to get in groups, you're going to work together, and you're going to have this scratch card. So each card has the different components and the enzymes that will be knocked down, and you have to scratch, and if you scratch in the right one, you get a point. If you scratch in the wrong one, you don't get any points. So let's see how many points you get. So sit down together, discuss it, and then you can text your points to the number I'm going to display to see how we did. So here are the spreadsheet cards. Okay, so let's see how we did in the scratching game. Well, the maximum amount of points you could get is 12, and I see, well, there's one group that got 10, one group that got 11, and two groups that got 12. That was great. So let's discuss what we would have expected. So anybody that got this got a 12. So let's think about it for a little bit. If we knock down drosha, which is here, we don't expect to affect the levels of the primary microRNAs, but everything else that is downstream will be downregulated. However, the target gene, we expect it to be upregulated due to the fact that if we have less microRNAs, and microRNAs downregulate genes, this is the effect that most likely will happen. And then when we knock down dicer, we're one step downstream, so we expect to have equal amounts of primary precursor, but less of the mature microRNA, therefore high expression of the genes. And when we only knock down argonaut, everything else in the process will be unchanged, but the target gene expression, we expect it to be high because argonaut forms the complex that downregulates gene expression. So good job to the teams that got the highest points. Let's discuss a little bit about how argonaut and the microRNA target these genes, how they recognize them, and how they repress expression. So this is how we would normally find the mRNA in the cell that we discussed in previous classes. When the translation is happening, the mRNA is forming a circle that is a result of an interaction of several transcription factors together with a poly-A binding protein. The ribosome will recognize sequences in this area, and it will go in the loop over and over again. However, when we have the RISC complex here with argonaut and the microRNA binding to the 3' untranslated region, when the ribosome is coming here, it's going to find something in the way. How does this happen? How does this binding happen? Well, only one of the two strands of the microRNA is able to bind. You know how RNA and DNA interact by complementarity of sequence. So we call that one strand of the microRNA the leading or the guide strand. And you can see here that even though the microRNA is 21 to 25 nucleotides, there's only a little portion of it that's binding to the 3' UTR. And it is required that only 7 nucleotides, at least, are complementary to the binding sequence. And that is what is called the seed region, or the seed sequence that some of you mentioned when you read the paper that was an assignment. So when this is happening, there are two ways that the microRNA can affect expression. First of all the microRNAs will be subjected to a process that's called deadenylation that is mediated by the microRNA and the RIS complex, and a series of enzymes will chew the poly-A tail. And then it could be either that the ribosome cannot reinitiate, as I mentioned before, it enters here, gets here, and it cannot restart. So we get decreased amount of protein, but not necessarily changes in the mRNA levels. But on the other hand, what can happen is that the RIS complex can recruit RNases that will come and chew this RNA. It could get rid of the cap structure that usually protects them from RNases, and will result in having less amount of mRNA as a result of the microRNA interaction. Therefore, there's less protein as well. So how do we study microRNAs in the lab? As I mentioned, there are more than 2,000 microRNAs, and those target thousands of genes. So we cannot just go and say, I think that microRNA 200 will target fuse. We actually have tools online that we can use that will look at the sequence of the microRNAs that are known, and will look at the sequence of the genes that they can target, and they have algorithms to determine whether the interaction is going to happen or not. These are some of the most commonly used target prediction tools. The one that's most commonly used is target scan. In there you put the gene, and it tells you the list of microRNAs. It tells you how many nucleotides of the seed sequence are conserved, and you can compare among species. And then once we identify microRNAs, we can actually validate these predictions. The fact that the computer is telling it's likely to bind doesn't necessarily mean that it's binding. So we can use something called the reporter assay, in which we can test the likelihood of a microRNA binding and affecting gene expression. And we can also use this targeting of genes. Knowing that a microRNA may bind, we can actually transfect the cell with that microRNA and check the expression of that gene. So the last thing we're going to discuss today is one of those that I just mentioned, the reporter assay. The reporter assay is a very clever way to study microRNA function. And what it consists on is we have a gene, we identify the 3' UTR, and we clone it in a vector downstream of a reporter gene, which could be luciferase in this case. So if a microRNA can bind to the 3' UTR, it can potentially repress the expression of luciferase. So this is a comparison of whether the microRNA will not bind or will bind. When there's no binding, luciferase will normally be expressed in the cells that are transfected with this vector, and you will see light after lysing the cells. However, if we have a microRNA affecting this expression, luciferase levels are going to be lower, either at the mRNA or the protein level, and there's going to be no light. And in the paper we discussed last week and this week, the investigators did that to test the effect of that single nucleotide polymorphism in the 3' UTR of the fused gene. So what they did here is they have a vector that has a luciferase, and this is the wild type sequence that contains a gene, normally, and this is where they predict that microRNA 141 or microRNA 200 may bind. Then they do one thing in which they delete the seed region. So all these nucleotides are taken away. And the next thing they do is they just switch the G for the A, as it's found in these patients that have ALS and increased fuse. So the next thing to do will be to transfect these vectors in cells together with the microRNAs that are being tested. So let's look at the results. This is the wild type vector, in which the normal 3' UTR is there, and we expect these microRNAs to bind. So what's happening here is that when we transfect the cells with microRNA 141, there's a decrease in expression. That means that the microRNA is binding to this region, and luciferase is less, as I explained in the previous slide. The same happens with microRNA 200. However, what is happening here, when we remove the seed region for the microRNA 141 and microRNA 200, we actually abolish the effect when we co-transfect with these two microRNAs. And not only that, just the fact that we change a single nucleotide in that seed region is sufficient to trigger the same effect. That's telling us that even though the seed region is important, those 7 nucleotides, just a change in a single nucleotide in a region that we thought it wasn't important for the gene, just the 3 UTR, is able to increase protein levels in these patients. So, now that we saw a little bit about how to work with microRNAs, and as all of you have expressed in the past that you want to work in something related to cancer, we're going to do an activity as an assignment. So you're going to get divided in groups, and each group is going to get a gene. All these 3 genes are involved in cancer, BRCA1, BRCA2, and P53. What I want you guys to do is to go in a gene bank and find the 3' UTR sequence for the gene that is assigned to you, and then use any of the online softwares I mentioned in the lecture and predict the binding of microRNAs. Once you do that, I want you to design an experiment. How would you test one of the microRNAs you found? And then predict the results of your experiment. So what would you see in your experiment if there is binding? And what would you see in your experiment if there is no binding of these microRNAs? So you have a week to get together in groups and do this assignment. And I hope you learned about microRNAs today and you enjoyed the lecture. And if you have any questions, we can talk after class. Thank you very much.